Plants prefer cross-pollination than self-pollination. So Hisardale is obtained by crossing Bicanary eaves with merino rams. The ancestors of modern-day frogs and salamanders are coelacanth. Biosphere reserve is an in-situ conservation where the plants and animals are preserved in large areas. They are conserved in large areas. Hello dear students, welcome back to Karnataka CET 2015 analysis. Hope you remember I keep telling that cracking this competitive exams needs more of your presence of mind, and also you should be thorough with the concepts of the subject. Then definitely it will not be a problem for you all to crack this Karnataka CET. So that is, today I'll be analyzing the Karnataka CET 2015 biology paper. Right. Let me get back to the first question. Moving on to question number 1. Which vector can clone a small fragment of DNA? Option A, bacterial artificial chromosome. Option B, yeast artificial chromosome. Option C, plasmid. Option D, cosmid. What is a bacterial artificial chromosome? It is a DNA construct based on the functional fertility plasmid. It is not a vector which is going to clone a small fragment of DNA. B, yeast artificial chromosome are genetically engineered chromosomes derived from yeast, Saccharomyces cerevisiae. Plasmid is a vector which can clone a small fragment of DNA. Cosmid, hybrid plasmid that contains a lambda phase. So which vector can clone a small fragment of DNA? Plasmid is a vector which clones only a small fragment of DNA. Why? Because it is small in size and is double-stranded. Why plasmid is used as a vector which can clone a small fragment of DNA? Because it is small in size and is double-stranded. So option A is wrong. Option B is wrong. Option C is correct. Plasmid is a vector which can clone a small fragment of DNA. Option D, hybrid plasmid that contains a lambda phase is wrong. So this is A is wrong, B is wrong, D is. So option C is correct. Option C is correct. Moving on to question number 2. Continued self-pollination results in. I think we all know that plants prefer cross-pollination than self-pollination. Continued self-pollination results in what? Reduced fertility and even production of the plant. So continued self-pollination results in option A, inbreeding depression. Option B, self-incompatibility. Option C, formation of unisexual flowers. And option D, gametes lose vigor. Continued self-pollination results in inbreeding depression. Inbreeding depression. What is A? Inbreeding depression is the reduced biological fitness in a given population as a result of inbreeding or breeding of related individuals. Same individuals. That means it is like self-pollination. Option B, self-incompatibility. Compatibility, adjustment. Self-incompatibility, not adjusting. Mechanism that prevents. So what is self-incompatibility? Mechanism that prevents pollen from one flower from fertilizing the other flower of the same plant. So it is like avoiding what? Self-pollination. Avoiding self-pollination. Avoiding self-pollination. Then formation of unisexual flower is also one method to avoid self-pollination. If it is a bisexual flower, chances of self-pollination is quite possible. As I said, Plants prefer cross-pollination than self-pollination, and they come out with certain outbreeding devices to avoid self-pollination, because continued self-pollination results in what? Inbreeding depression. Gametes lose vigor. This is also one strategy to avoid what? Self-pollination. To avoid self-pollination. To avoid, gametes will lose their vigor or functionality. Avoid self-pollination. So, continued self-pollination results in inbreeding depression. That is, option A is correct. So, option B is wrong. Option C is wrong. Option D is wrong. So, option A is what? Inbreeding depression. So, continued self-pollination results in inbreeding depression. Moving on to question number 3. Identify the wrong statement. Alleles IA and IB produce sugars. Both IA and IB are present together and they express because of codominance. Alleles B and C also produce sugars. Option D, when IB and small b or I are present, only IB is expressed. As we know that the plasma membrane of the RBC has sugar polymers that are going to protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has 3 alleles, that is IA, IB and small I. The alleles IA and IB produce a slightly different form of sugar, that is IA and IB are going to produce sugar while small I does not produce any sugar. So option A, alleles IA and IB produce sugar, is wrong. Then both IA and IB are present together and they express because of codominance, wrong. Alleles B and C produce sugar. Alleles B and C produce sugars. When IB and B are or a small I are present, only IB is expressed, is wrong. So option C is correct. Moving on to question number 4. The codon AUG. AUG is a triplet codon. Has dual function, double function. It is an initiation codon and also codes for. It is also called as an initiator codon. So, the protein synthesis will start with an initiator codon. So, AUG is an initiator codon. Options: A. Formaldehyde, B. Methionine, C. Phenylalanine and D. Serine. All are different types of, and this is not an amino acid. All 3 are amino acids. So, during translation, AUG acts as an initiator of protein synthesis. So, double function. Initiator of protein synthesis, as well as it is a codon coding for an amino acid called methionine. Short form is what? MET. So, it is coding, it is an initiator. So, whenever a protein molecule is formed, it comes first. So, what is this? This is MET, methionine. So, next comes next amino acid. So, like this, it goes on. So, finally, when complete protein molecule is formed, it spirals like this. Right? So, then it is called as a protein molecule. It will twist like this and forms a protein molecule. So, always this is an initiator codon. There are initiator codons and terminator. Terminator codons will stop the protein synthesis. Initiator will start the synthesis of protein. So, AUG has dual function. So, AUG stands for an amino acid called methionine. Short form is MET. So, A. Formaldehyde wrong. B. Methionine is correct. C. Phenylalanine is wrong. D. Serine is wrong. So, option B is correct. Option B is correct. Moving on to question number 5. Natural killer lymphocytes are an example for A. Cytokine barrier, B. Physiological barrier, C. Physical barrier and D. Cellular barriers. As we know that natural killer lymphocytes that is we commonly call it as NK cells, act like independent single-celled organisms. That is, they act as what? Cellular barriers. They act as cellular barriers and they form what? Second line of defense. Second line of defense. So that and natural killer lymphocytes are an example for cellular barriers, which is going to act as what? Second line of defense. Option A, cytokine barriers are like interferons, tumor necrosis factors, etc. Physiological barriers, it may be the body temperature or pH of body fluids. Physical barriers might be the skin, mucous membrane, etc. Cellular barriers, as I said, lymphocytes, then macrophages, neutrophils. So, natural killer lymphocytes are an example for cellular barriers. So, option A, cytokine barriers is wrong. Option B, physiological barrier is wrong. Option C, physical barrier is wrong. Option D, cellular barrier is correct. So, option D, natural killer lymphocytes are an example for what? Cellular barriers. Moving on to question number 6. Identify the phylum X. Hope you all know that there are 9 invertebrate phyla. In that, kingdom Animalia. What will be the tissue grade? Means what? Cells are there and cells are there and cells group into tissues. There are 4 levels of body organization: cellular level, tissue level, organ level and organ system level. So, the animals which are restricted only to tissue grade of level, that is bilateral symmetry, means what? Body can be cut into 2 equal halves in one plane or one direction is called bilateral symmetry. Acoelomates means what? Without coelom. So, cells with tissue grade of body organization, body can bilateral, then acoelomates. X. So, what is this X? All these features belong to which phylum? Which phylum? Option A, Aschelminths? No. B, Ctenophora? No. C, Hemichordata? No. D, Platyhelminths. All these are the characteristic feature of phylum Platyhelminths. That is flat worms. So, option D is correct. Option D is correct. So, X is phylum Platyhelminthes. Phylum Platyhelminthes. Moving on to question number 7. With respect to eichhornia, statement X, it drains off oxygen from water and is seen growing in standing water. Statement Y, it is an indigenous species of our country. Eichhornia, eichhornia is called water hyacinth. You know, this is called as a terror of Bengal. Terror of Bengal, and this eichhornia is an invasive weed which is going to spread all over the water body and it is going to drain out the oxygen content from the water, thereby killing the aquatic animals. So, it drains off oxygen from water and is seen growing in standing water is correct. It is an statement Y. It is an indigenous species of our country. It is wrong. Why it is not? It is not a native variety. It is an exotic species, or it is an invasive weed, and it is a native of South America, not our country. It is a native of South America, first introduced in Egypt and then in South Africa in 1950, and it was brought to India because of its as an ornamental plant, showy plant, ornamental plant, because of its beautiful flowers and for the leaves. So it was brought to India because of its beautiful flowers and the leaves. So statement Y is wrong. Statement X is correct. Statement Y is wrong. Option A. Both the statements X and Y are correct. Wrong. B. Both statements X and Y are wrong. Is wrong. C. Only statement X is correct. Correct. And Y is wrong is correct. Option D. Only statement Y is correct and X is wrong is wrong. So option C is correct. That is, statement X is correct and Y is wrong. Moving on to question number 8. Seeds without fertilization is obtained from option A. Parthenocarpy. What is parthenocarpy? Development of fruit without fertilization. Option B. Apomixis. What is apomixis? Seed formation without fertilization. Polyembryony, formation of more than one embryo. Option C. Dormancy, it is a suspended phase where all the activities are slowed down. Slowed down for a period of time. The seeds will undergo dormancy or the bud will undergo dormancy, all that. So, seeds without fertilization is obtained from option A. Parthenocarpy, wrong. Option B. Seed formation without fertilization, correct. Apomixis. Polyembryony, formation of more than one embryo, wrong. Option D. Dormancy is wrong. So option B is correct. That is, seeds without fertilization is obtained from apomixis. And such seeds are called as what? Apomictic seeds. Apomictic seeds. Right. Question number 9. The hormone which acts on Sertoli cells and stimulates the process of spermiogenesis. What is spermiogenesis? Transformation. Spermatid into motile sperm. The hormone which acts on Sertoli cells and stimulates the process of spermiogenesis. Androgen A. Option A. Androgen is a male hormone which is responsible for formation of male secondary characters, sexual characters. Formation of male secondary sexual characters. B. LH. LH is important for what? Spermatogenesis. GnRH, which is produced from the hypothalamus. Hypothalamus produces what? Two types, that is FSH and LH. Option D, FSH, is responsible for spermiogenesis. FSH and LH both are hormones of GnRH, gonadotrophic releasing hormone, which is very important both for males and females. Both for males and females. So, the hormone which acts on Sertoli cells and stimulates the process of spermiogenesis. Androgen, option A is wrong. B. LH is wrong. C. GnRH is wrong. So, D. FSH is correct. That is, the one which brings about spermiogenesis is FSH. So, option D is correct. Moving on to question number 10. The nitrogen base found only in DNA is called. In DNA has 4 nitrogen bases. That is adenine, guanine, cytosine and thymine. Right. So the nitrogen base found only in DNA is also called. Thymine of DNA is also called as 5-methyl-uracil. And it is specifically found where? Only in DNA. So the thymine of DNA is replaced by uracil of RNA. So in DNA it is thymine and in RNA it is uracil. So it is also called as what? 5-methyl-uracil. Option A, 5-methyl-uracil. Option 5-methyl-uracil is nothing but what? Thymine. Thymine is 5-methyl-uracil. Ammonium chloride is not a nitrogen base. Uracil, as I said, it is found only in RNA. Guanine is both. Here in RNA we come across adenine AGCU. So guanine is common for both DNA and RNA. So the nitrogen base found only in DNA is also called 5-methyl-uracil is correct. B, ammonium chloride is wrong. C, uracil is wrong. D, guanine is wrong. Moving on to question number 11. Hisardale is obtained by crossing. Hisardale is a new breed which is obtained by cross hybridization. New breed of what? Sheep developed in Punjab, crossing between Bicanary Eves and Merino rams. So Bicanary Eves and Merino rams. It is obtained by crossing through recombination by two different species. Recombination of two different species. So Bicanary Eves and Merino rams. So Hisardale is obtained by crossing Merino Eves with Bicanary. It is Bicanary Eves with Merino Eves. So it is wrong. Option B, Bicanary Eves with Merino rams gives what? Hisardale. So it is correct. Option C, horse with donkey is called what? Mule. Which is more economically, commercially a very important animal. Which is stronger, which can do hard duty, all that. So horse with donkey is mule. This is specific hybridization. Superior bull with superior cow. That is, which produces superior progeny. Option D is wrong. Option C is wrong. So option B is correct. So Hisardale is obtained by crossing Bicanary Eves with Merino rams. Moving on to question number 12. The ancestors of modern day frogs and salamanders are. All of us know evolution brings what? Gradual morphological changes. The discovery of a low fish called coelacanth, they were the first amphibians that have evolved from fish-like organisms. Coelacanth or the low fish have evolved from fish-like organisms. So, low fish or coelacanth were ancestors of modern day frog and salamanders. Option A, jawless fish means what? Agnatha, without teeth, like lampreys, hagfishes. Coelacanth, it is an ancestor of modern day frog and salamanders. Ethiophis, limbless amphibian. Limbless amphibian. Amphioxus, it's a cephalopodate. Example for cephalochordates. So, the ancestors of modern day frogs and salamanders are option A, jawless fish, wrong. Option B, coelacanth. Coelacanth is also called as what? Low fish. Then Ethiophis, C, wrong. D, Amphioxus is wrong. So, option B is correct. Coelacanth. The ancestors of modern day frogs and salamanders are coelacanth, which are also called as what? Lobe fish. Moving on to question number 13. During sewage treatment, biogas produced includes. Option, what is biogas? Biogas production reduces what? The pollution potential in the wastewater by reducing the oxygen levels required by the organic matter, like it will be like CH4, that is methane, H2S hydrogen sulfide, and CO2 is carbon dioxide. So the biogas which is produced is what? Methane, hydrogen sulfide and carbon dioxide. Option A, methane, oxygen and hydrogen sulfide. Option, because here it is oxygen, wrong. Methane, hydrogen sulfide and carbon. Hydrogen sulfide, methane, sulfur oxide, wrong. Option C, hydrogen sulfide, nitrogen, methane, wrong. Option D, methane, correct, hydrogen sulfide, correct, and carbon dioxide, correct. So, option D is correct. So, during sewage treatment, Biogas produced includes what? Methane, hydrogen sulfide and carbon dioxide. So option D is correct. Moving on to question number 14. If 30 joules of energy is trapped at producer level, then how much energy will be available to peacock as food in the following chain? Plant to mice to snake and peacock. According to Lindman law, which is also called as 10% law, as we know, the energy is transferred from one level, trophic level, to another trophic level. During the transfer of energy from one trophic level to the next, only 10%, that's why it's called as what? 10% law. Only 10% of energy is passed to the next trophic level. So only 10% of energy. For example, if a plant traps 30 joules of energy, of this only 10%, that is 3 joule, is getting absorbed by mice. So only 3 joule is getting absorbed by mice. From mice, only 10%, that is 0.3 J, is used by snakes. So 0.3 joules is used by the snake. From snake, only 10% which is available goes to the peacock, which is around 0.03 joules. So if 30 joules of energy is trapped at producer level, then how much energy will be available to peacock as food in the following chain? It will be 0.03 joules. Option A, 0.03 joules, correct. Option B, 0.003 joules is wrong. C, 0.3 joule is wrong. And D, 0.003 joule is wrong. So option A is correct. Option A, how much? So the energy that is available to peacock is how much? 0.03 joules. Moving on to question number 15. Which one of the following is not an ex-situ conservation? Conservation is of two types. One is called in-situ and the second one is called ex-situ conservation. Zoo, then botanical garden, all cryopreserve bank, all these are examples for ex-situ. In-situ is national parks, then biosphere reserve. All these are the examples for in-situ conservation. Which of the following is not an ex-situ conservation? Seed bank is an ex-situ conservation, where storing of seeds for what? For genetic diversity. B, botanical garden is what? Ex-situ conservation. Cryopreservation is what? Process of cooling and storing cells and tissue at low temperature to maintain their viability, functionality. Biosphere reserve is an in-situ conservation, where the plants and animals are preserved in large areas. They are conserved in large areas, right? So option A, seed bank, is wrong. Option B, botanical garden, is wrong. It's not an ex-situ conservation. It is an ex-situ conservation. C, it is an ex-situ conservation. D, biosphere reserve is not an ex-situ conservation. So option D. Which of the following is not an ex-situ conservation? It is biosphere reserve. Question number 16. One hormone hastens means fastens. Fastens the maturity period in juvenile conifers. A second hormone controls xylem differentiation, while the third increases the tolerance of plants to various stresses. They are respectively. Option A, auxin, gibberellin, cytokinin. Option B, auxin, gibberellins and abscisic acid. Then option C, gibberellins, auxins and cytokinins. Gibberellin, auxin and abscisic acid. Moving on to gibberellin. Gibberellin is mainly responsible, it's a phytohormone or a plant hormone which is responsible for stem elongation, leaf senescence and also hastens what? Maturity period in juvenile young conifers. Auxin is a growth promoting hormone, controls xylem differentiation, whereas abscisic acid, which is also called as stress hormone. This is also called as stress hormone, increases the tolerance of plants in various stress conditions. So the hormone which hastens maturity period is gibberellin, and the second hormone, this is the first hormone which hastens the maturity period in juvenile conifers. The second hormone controls xylem is auxin. And the third one which increases is abscisic acid, increases tolerance. So gibberellin, auxin, abscisic acid. Option A, auxin. So first only auxin is there, it is wrong. B, auxin is there, this is also wrong. Here gibberellin is correct. Option D, gibberellin is correct. Second one is auxin is correct. Auxin is correct. But third one is what? Abscisic acid. Here it is cytokinase. See, option C is wrong. D, gibberellin, auxin, abscisic acid is correct. So option D is correct. Moving on to question number 17. The element responsible for the ring structure of chlorophyll and maintenance of ribosome structure is magnesium. Potassium is very much B, potassium, C, calcium, D, sulfur. Magnesium. Magnesium helps maintain the structural integrity of the chlorophyll pigment, and it forms the center of porphyrin ring of the green plant pigment called chlorophyll. Also helps convert light into usable form of energy. And magnesium plays an important role in joining the two pigments, plays an important role in joining the two fragments of ribosomes during the process of translation. Potassium is very much important for closing and opening of stomata. Closing and opening of stomata. And this is calcium is important for plant growth and nutrition. Whereas sulfur is important to form some enzymes. Helps in formation of some enzyme. So, the element responsible for the ring structure, porphyrin ring structure of chlorophyll and maintenance of ribosome structure. The element which is responsible is what? Porphyrin ring structure is magnesium. Then also plays an important fragment of what? Ribosome. Ribosome structure. So magnesium is correct. So option A is correct, B is wrong, C is wrong, and D is wrong. So option A is magnesium. Moving on to question number 18. Which of the following sentences is correct? Cells of all living organisms have a nucleus. B, both animal and plant cells have a well-defined cell wall. C, in prokaryotes there are no membrane-bound cell organelles. Cells are formed from abiotic materials. Option A, cells of all living organisms have a nucleus is wrong. It is the wrong sentence because all the cells of living organisms will not have nucleus. Then B, both animal and plant cells have a well-defined cell wall. Wrong. Animal cells do not have a cell wall. Wrong. In prokaryotes, there is no membrane-bound organelles. Correct. Membrane-bound organelles are present only in eukaryotes. Present only in. So here, in prokaryotes, there are no membrane-bound organelles. Correct. Moving on to D, cells are formed by D from abiotic are wrong because cells have the living substance called protoplasm. So option D is wrong. So option C is correct. Which of the following sentence is correct? In prokaryotes, there are no membrane-bound cell organelles. Moving on to question number 19. Label the correct parts of the Mysin monomer. This is the picture of a Mysin monomer. We need to label A, B, C, D. Each myosin, we call it as thick filament, myosin filament. Myosin molecules are composed of what? Head, neck and tail. So A is head. Then B is cross arm. Then C is what? Actin binding sites. Actin binding site. Then D is ATP binding site. So A is head, B is cross arm, C is actin binding site, and D is ATP binding site. Option A is cross arm. Here only it is wrong. A is head correct. Here it is A actin binding site wrong. Here also ATP binding site is wrong. First answer only is wrong. Only here you can see head. Nowhere in the three options you find head. So B is cross arm correct. C is actin binding site correct. D is ATP binding site is correct. So option B is correct. So A is head, B is cross arm, then C is actin binding site, and D is what? ATP binding site. So option B is correct. Option B is correct. Moving on to question number 20. The 2000-year-old seed excavated from King Herod's palace at Dead Sea belonged to Lupine articus, which is excavated from Arctic tundra, and the seeds were viable for the seeds germinated after what? 10,000 years. Strobilanthus kuntiana flowers once in 12 years. Dendrocalamus, it is a male bamboo. Phoenix dactylifera, that we call it as date palm. During excavation at the site of Herod the Great Palace in Israel in the early 1960s, archaeologists unearthed a small stockpile of seeds stored where? In clay jar. Imagine those years, the clay was quite commonly used, dating back to 2000 years. And the seeds were of what? Phoenix dactylifera, which we commonly called as date palm. So the 2000-year-old seed excavated from King Herod's palace at Dead Sea belonged to what? Phoenix dactylifera. A, 10,000 years, wrong. Which flowers? Wrong. Then this is male bamboo, wrong. This is correct. So option D is correct.